Adjuvants. Most proteins are poorly immunogenic or non-immunogenic when administered by themselves. Strong adaptive immune responses to protein antigens almost always require that the antigen be injected in a mixture known as an adjuvant. An adjuvant is any substance that enhances the immunogenicity of substances mixed with it. Adjuvants differ from protein carriers in that they do not form stable linkages with the immunogen. Furthermore, adjuvants are needed primarily for initial immunizations, whereas carriers are required to elicit not only primary but also subsequent responses to haptens. Adjuvants can enhance immunogenicity in two different ways. First, adjuvants convert soluble protein antigens into particulate material, which is more readily ingested by antigen-presenting cells, such as macrophages. For example, the antigen can be absorbed on particles of the adjuvant, such as alum, made particulate by emulsification in mineral oils, or incorporated into the colloidal particles of ISCOMs. This enhances immunogenicity somewhat, but such adjuvants are relatively weak unless they also contain bacteria or bacterial products. Such microbial constituents are the second means by which adjuvants enhance immunogenicity, and although their exact contribution to enhancing immunogenicity is unknown, they are clearly the more important component of an adjuvant. Microbial products may signal macrophages or dendritic cells to become more effective antigen-presenting cells. One of their effects is to induce the production of inflammatory cytokines and potent local inflammatory responses. This effect is probably intrinsic to their activity in enhancing responses, but precludes their use in humans. Nevertheless, some human vaccines contain microbial antigens that can also act as effective adjuvants. For example, purified constituents of the bacterium Bordetella pertussis, which is the causal agent of whooping cough, are used as both antigen and adjuvant in the triplex DPT. And that is adjuvants.